Hi everyone, welcome back to another video on Campbell Biology, where today we will be talking about soil and plant nutrition, or chapter 37 in the textbook. And before we begin, I would first like to thank all of the people that subscribed, the entire 10 to 12 that are subscribed, and those that are currently watching. Your time is greatly appreciated and we thank you for it. And I will say that all of our diagrams are from Campbell Biology unless otherwise stated, and that in general, our videos will be structured with the lesson in the beginning and some practice problems in the end going into the future. With all that being said, let's begin today's topic. So, as discussed in the last couple of chapters, plants obtain nutrients from both the atmosphere and the soil. By using sunlight as an energy source, they produce organic nutrients by reducing carbon dioxide to sugars through, as most of you know, photosynthesis. They also take up water and various inorganic nutrients from the soil through their root systems. And this chapter focuses on the process of these gaining these nutrients, the study of the minerals necessary for plant growth. And so let's move on to the layers of the soil. And so the upper layers of the soil, specifically the around this part, around this part are where plants absorb nearly all the water and minerals they require, and it contains a wide range of living organisms that interact with each other and with the physical environment. And before we begin talking about soil layers or soil horizons, as you see there, I would like to talk a little bit about soil particles. And basically, soil particles can range from anywhere between 0.02 to 2 millimeters diameter, which is something similar to coarse sand, like sand at the beach. And then as we go lower in diameter, we go to silt at 0.002 to 0.02. And you get the drill, and eventually we go to clay particles, which are less than 0.002. And now, mineral particles released by weathering become mixed with living organisms and humus, the remains of dead organisms, and other organic matter, forming topsoil. The topsoil and other soil layers are called soil horizons. So, as you can see here, we have three main horizons: the A horizon, the B horizon, and the C horizon. And so they're in order by depth. So what that means is that this is the A horizon. That this middle layer is kind of like the B horizon. And then finally, this layer of basically rock and little to no organic material is the C horizon. And so the important features of each is that A horizon contains the most organic matter by far. It's also known as the topsoil. And it contains a mixture of broken down rock of various textures, living organisms, and decaying organic matter. And then the B horizon is sort of like a middleman. It's kind of like a little bit of the organic matter, but much less than A, and then it's also less weather than A. But it's mostly rock at this point. And then C is all partially broken down rock. And some of this rock will serve as parent material for minerals that help form the upper horizon. So minerals like your Ca2+, Mg2+, they can all come from the C horizon. Now, the topsoils that are the most fertile, so let's focus on this A horizon because that's where most of the magic happens with soil, right? The most fertile topsoils are loams, which are composed of roughly equal amounts of sand, silt, and clay. And so the reason why we want them all to be sort of in equal amounts is because loamy soils are a 1-to-1-to-1 ratio of sand, silt, and clay. And this is because if there's too much sand, the soil has too much internal space for roots to properly adhere to the soil. And if there's too much clay, that means that the soil will retain too much water and the plant won't be able to get any. And so that's why we want equal parts of sand, silt, and clay. Now, one thing about topsoil is that it has many soil particles in it that are negatively charged. So as you can see in this diagram, which we'll get to the process later, you can see that the soil particle is surrounded by negative electrons. And therefore, since most of the soil particles are negatively charged, the plant nutrients nitrate, phosphate, and sulfate are all easily lost by leaching, where water flows through the soil, and because of the water's partial positives, on the H, they're really easily attracting the negatively charged anions, the nitrates, phosphates, and sulfates, and they'll bring them more down into the soil. So as water flows through the soil, those nutrients will also go down, and thus the roots would be able to less easily reach these nutrients, and thus these nutrients are easily lost by leaching. And so one thing about leaching is that it doesn't really happen to positive cations, because like we said, because soil particles are negatively charged, then that means that cations like K plus, Ca2 plus, and Mg2 plus are all going to attract to these negative soil particles, and so they're not lost by leaching as easily as the negative nutrients. But roots, however, they don't absorb mineral cations directly from soil particles. They absorb them from the soil solution. Mineral cations enter the soil solution by cation exchange, which is this process in figure 37.3 from chapter 11th edition of Campbell Biology. And cation exchange is a process in which cations are displaced from soil particles by other cations, like the hydrogen cation. So as you can see here, roots acidify the solution by releasing carbon dioxide, and thus this causes a influx of hydrogen protons to enter the soil. And because the reason for these hydrogen ions is because when water reacts with carbon dioxide, the resulting carbonic acid will dissociate into the bicarbonate ion and the hydrogen proton. And these hydrogen protons will neutralize the negative charge of soil particles. So think of them as like crowding out the available negative spaces for the Mg2 plus, Ca2 plus, and K plus to be absorbed by this, attracted to the soil particles. So imagine like there's a H plus here blocking it out, and so therefore the Mg2 plus will go to the slightly less negative, more negative thing, the root hair. And so that's how the plant is able to absorb nutrients through cation exchange. And the soil's capacity to exchange cations is determined by the number of cation adhesion sites, so all these little spaces here, and by the soil's pH, which is the next slide. And because the clay particles have high surface area to volume ratio, they will allow for ample cation binding. So therefore, as your organic matter and clay concentration increase, your soil pH will on average be, or sorry, not your pH, but the cation exchange capacity will also increase because of that high surface area to volume ratio of clay content. So now let's move on to, again, well, we discussed it here, but let's just write it again for sake of clarity. So as in general, as organic matter and clay increase, the cation exchange capability will also increase. And that is because, well, the clay content has obviously clay particles, and because these clay particles have high surface area to volume ratio, they allow for ample cation binding. And so you can see here in the soil particle that there's a lot of spaces, a lot of areas for these cations to bind to, and that's again because of the clay particle's high surface area to volume ratio. And thus, by the soil's pH, this will naturally cause a neutralization of the soil pH, I think. But let's move on to more general soil pH. Soil pH is an important factor that influences mineral availability. As you can see here, it entirely depends on the concentration of hydrogen protons. When hydrogen proton concentration increases, the pH decreases. And so therefore we're more able to take in nutrients or the released cations from the soil particles. And most plants, in fact, prefer slightly acidic soil because the high H plus concentrations can, like we said, displace positively charged minerals like Ca2 plus, K plus, and Mg2 plus from the soil particles. If it's too alkaline, what do? We can add sulfate, SO4 2 minus. If it's too basic, so that means if a pH is too high. If pH is too low, or if it's too acidic, then we'll add lime, which is in the form of CaOH2. Or in general, you just need some sort of OH in this OH minus to enter the solution, to enter the soil solution, the soil system, and increase that pH by decreasing hydrogen concentration. Now, when the soil, one thing to note, let's erase all of this. One thing to note is that when the hydrogen ion concentration is less than 10 to the negative, is greater than 10 to the negative 4 molar, that toxic aluminum ions, Al3 plus, become more soluble and are absorbed by roots, and these stunt root growth and prevent the uptake of calcium, a needed plant nutrient. Some plants can cope with high Al3 plus levels by secreting organic anions that bind aluminum 3 plus, but especially in tropical regions, this is still a really persistent issue. And so, it should be 10 to the negative 5. But in general, when pH, again, like we said, when pH is less than 5, then aluminum ion solubility increases. And that's a problem because it displaces space needed for the calcium 2 plus, which is an essential plant nutrient. And now the inorganic substances in plants contain more than 50 chemical elements. And thus, therefore, we must distinguish elements that are essential from those that are merely present in the plant. And so, therefore, a chemical element is considered an essential element only if it is required for the reproduction and completion of the life cycle. So I'll say that again because I think this might be important. Chemical elements are considered essential nutrients only if it's required for reproduction and life cycle completion. And so therefore, that is how we will distinguish essential and non-essential nutrients. But before we dive deeper into that, I would like to talk about a little bit of how this applies to agriculture, specifically in the form of phytoremediation. Phytoremediation is the process by which you repair the soil, sort of. So in this plant, its name is Alpine pennycress. And what this plant does is that it can tolerate more than 300 times the normal zinc 2 plus capacity as most plants. And so relatively, in other words, it has a 300 times higher tolerance for zinc 2 plus than other normal plants. And what phytoremediation in this situation looks like is that the Alpine pennycress will take up most of that Zn 2 plus. So let's draw it out. Let's say we plant in Alpine pennycress here and here. They're just going to start taking up all these zinc 2 plus ions. And then eventually, Once they're done, once these are crossed out, these plants are going to be cut off and they're going to be removed and then disposed of safely. Now, let's get back to the main topic, which are our nutrients. To determine which chemical elements are essential, researchers will use hydroponic culture, where plants are grown in a mineral solution instead of soil. So, how you would test for if a plant if a plant nutrient is essential, is you would do a control solution with all the minerals, and then one without, let's say, iron. And so you see here, iron is an essential nutrient because its deficiency stunts the plant's growth. Now, such studies have helped it identify 17 essential elements needed by all plants, which are 9 macronutrients and 6 of these are the major components of organic compounds forming a plant's structure. And before we get into that, 9 of these are called macronutrients because plants require them in large amounts. That does not mean that without some micronutrient, that does not mean that they are more necessary than micronutrients, however. Many of these micronutrients function as cofactors in the citric acid cycle, and as well as playing a role in other areas of the plant. And what is important to note here is that a deficiency of a micronutrient can just as easily kill a plant. So micronutrients are equally important to macronutrients. And so 6 of these macronutrients, let's go into that, are the familiar or major components of organic compounds, which are the carbon, oxygen, hydrogen, nitrogen, phosphorus, and sulfur, which are the major like we said, the major components of organic compounds. And the other 3 nutrients are potassium, calcium, and magnesium. Out of all the mineral nutrients, nitrogen contributes the most to plant growth. And plants require nitrogen as a component of proteins, nucleic acids, chlorophyll, and other important organic molecules. The next table will summarize the function of these macronutrients. The other essential elements are called micronutrients because plants need them in only tiny quantities. They are chlorine, iron, manganese, boron, zinc, copper, nickel, and molybdenum. And sodium is a 9th essential micronutrient for plants because that use the C4 or CAM pathways. So C4 and CAM are deviations from the traditional C3 photosynthesis. And those are a topic for another day. But the important thing is that Na+, or rather sodium we'll call it, will be in a 9th micronutrient for C4 and CAM plants. And now, deficiencies of different elements may have different symptoms. So before we get to that, I'll leave this table here. You can see that our CHOMPs are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur are the most important macronutrients. And then the potassium, calcium, manganese are the three inorganic, or rather not a major component of most organic compounds. And as for a table of micronutrients, you don't really need to know most of their like specific functions. Just know that they most of the time that they function as a cofactor or an electron carrier for the electron transport chain and the citric acid cycle. And again, micronutrients are important because they generally play catalytic roles that plants need in only tiny quantities. And the requirement for molybdenum, for example, well, let's take a comparison. Molybdenum is so modest that there's one atom per 60 million hydrogen atoms. Yet, a deficiency of molybdenum, like we've been discussing, can weaken or kill a plant because micronutrients are equally important as macronutrients. And so here are some common deficiencies. These are all from maize plants. Which you can see here that unhealthy one, there's no yellowing or chlorosis. And then if we go here, if we see nitrogen deficiency, we can look at our reference. Nitrogen, chlorosis at tips of older leaves and common in heavily cultivated soils. And we can clearly see here that there's a clear streak and there's obviously yellowing at the tip. Phosphorus deficient, let's look at our reference. Healthy appearance but very slow development and purpling of veins. And you can clearly see here that there's going to be purple. And finally, potassium deficient, we're going to see a drying of leaf edges. And we can clearly see here that there's a sort of desiccation going on here. Now, one thing that I'm going to talk about now is that are rhizobacteria. Rhizobacteria are bacteria that live either in close association with plant roots or in the rhizosphere, the soil closely surrounding plant roots. These rhizobacteria will form mutualistic associations with plant roots. They depend on nutrients such as sugars, amino acids, and organic acids that are secreted by plant cells. And in return, plants reap many benefits from these mutualistic associations. Some rhizobacteria produce antibiotics, others will absorb toxic metals or make nutrients more available. And still others convert gaseous nitrogen into usable forms. And in general, rhizobacteria, there are so many different types of rhizobacteria and plant interactions that almost 95% of all plants on Earth have some sort of mycorrhizal relationship. Some rhizobacteria are free-living in the rhizosphere, the surrounding area of the roots and soil, whereas other types of rhizobacteria are endophytes that live between cells within the plant. So we'll go to the nitrogen cycle next, but before that, let's talk about the legume mycorrhizal relationship. The relationship between rhizobium, these specific special bacteria, and legume roots involves dramatic changes in root structure. Along the legume's roots are swellings called nodules. So they look something like this. You're going to see that they have these really obvious globules or like globs within the roots. Those are the nodules. And these are composed of plant cells that are infected by rhizobium bacteria. And infected is the best word we can use, but it's not really appropriate in the sense of feeling because, infection is typically negative. But in fact, this is one of the most positive relationships in possibly all of biology. Inside each nodule, rhizobium bacteria assume a form called bacterioids, which are contained within vesicles formed in the root cells. So we can see here, this is an example of a bacteroid. And therefore, what these relationships generate is more usable nitrogen for plants than all industrial fertilizers used today, and at virtually no cost to the farmers of today. Nitrogen fixation by rhizobium requires an anaerobic environment, an environment without oxygen. It's in a condition facilitated by the location of the bacteroids inside living cells in the root cortex. The lignified external layers of root nodules help to limit gas exchange, and some root nodules appear reddish because of a molecule called legumoglobin. And you can see here that hemoglobin is much like sounds really similar to the protein complex that we use in our blood. And you're right, I'm pretty sure that this heme group or the whatever iron that contains this protein that binds reversibly to oxygen is very similar to the hemoglobin in our own red blood cells. Anyways, this red protein is an oxygen buffer, reducing the concentration of free oxygen and thereby providing an anaerobic environment for nitrogen fixation while regulating the oxygen supply for the intense cellular respiration required to produce ATP for nitrogen fixation. So let's look at the process here. Roots emit chemical signals that attract rhizobium bacteria, which emit signals that simulate root hairs to form and form an infection thread by an invagination of the plasma membrane. And so we can see here that the infection thread containing the bacteria penetrates the root cortex, which is what it's doing right here. Sorry, right here. And the cells of the cortex and pericycle begin to divide, and vesicles containing the bacteria bud into cortical cells from the branching infection thread. Bacteria within the vesicles develop into nitrogen-fixing bacteroids. So you can see here that the bacteroids are beginning to form, and then growth continues in these affected regions, and the two masses of dividing cells fuse and form the nodule. So remember these two will become one and form a whole nodule. And the nodule develops vascular tissue that supplies nutrients to the nodule and carries nitrogenous compounds into the vascular cylinder for distribution throughout the plant. That's how the plant gets these supply of nitrogen. The mature nodule grows to be many times the diameter of the root, and then this is the whole nodule that we've been showing here. This is going to be that big glob over here. And a layer of lignin-rich sclerenchyma, which are these pink cells, form and reduce the absorption of oxygen and thereby keep a anaerobic environment necessary for nitrogen fixation. And with that being said, let's go back to the nitrogen cycle. The nitrogen cycle involves all these components you see here, and there's not much to say about it except that it is one of many cycles within our our biological systems. We also have the carbon cycle, the water cycle, and even I think the phosphorus cycle, just to name a few. But the important thing you have to notice here is that the nitrification process is sorry, the nitrification is the biological oxidation of ammonia to nitrates. So you can see here that the ammonia will, through this nitrification process, will go to here, will form nitrate, which is then, again, like we said, a nutrient for plants. Excuse me, a nutrient for plants. It will be absorbed through the roots. And so there's several intermediaries, such as because first it has to become ammonium, which becomes nitrite, which then gets an oxygen added to become nitrate. Now, one thing that we will talk about is the types of mycorrhizae. And there are mainly two types of mycorrhizae, ecto and arbuscular. So let's talk about the ectomycorrhizae first. The ectomycorrhizae form a dense sheath or mantle of mycelia over the root surface. So you can see here, there's almost a sort of secondary layer here of ectomycorrhizae. And its fungal hyphae extend into the soil. And another thing is that the hyphae grow into the root cortex, but they do not penetrate root cells. So you can see here that they grow in like the cracks and crevices, but they don't penetrate into the root cell. They create a network in the apoplast, the extracellular space, which is labeled right here. So everything like in between the lines of the root cells, if you will, they will form a network in those to facilitate nutrient exchange. Plants with ectomycorrhizae will have thicker, shorter, and more branched roots compared to uninfected ones. They do not form root hairs, as fungal mycelium provides the extent of the surface area because fungal hyphae extend into the soil and increase surface area. They're found in about 10% of plant families, mainly woody species, which may include pine, oak, birch, and eucalyptus. And again, the main thing here is that mycorrhizae interactions are not limited to just legumes. There are many other more specific interactions that result in almost 95%, like we've been saying, 95% of plants forming mycorrhizal associations. Anyways, back to the point. We have another form which are called arbuscular mycorrhizae. As you can see here, you can immediately notice a few differences. They're embedded within the root rather than forming a sheath, so there's no sort of a secondary layer here. It's just sort of some branches here, some branches there, but never an aggregate. The microscopic soil hyphae grow toward contact and line along the root surface. So rather than extending outwards of the plant, they go to the plant. And hyphae penetrate between epidermal cells and enter the root cortex, where they digest small patches of cell walls. And you can see here that what's important here is they don't pierce the cell membrane, but instead they grow into invaginated tubes formed by the root cell's membrane. So you can see here they form into these little arbuscules, which comes from the I think represents little trees, and those are critical sites of nutrient exchange. Now, oval vesicles may form within hyphae, as you can see here. And what these do is, it's not very clear what they do, but it's surmised that they are for food storage. And these are by far the most common type. They are found in over 85% of plant species, including even most of the crops that we eat today, like maize and wheat. And again, like we said, only about 5%, because, well, this is 10% of families, and this is about 85% of plant families. So therefore, about 5% do not form any sort of mycorrhizal association. And now we're going to talk about some unusual plants. And what these do is that they get nutrients from away without soil, essentially. And so you can see here, this is a staghorn fern, which is an epiphyte. And what this does is that rather than, epiphyte stands for, I'm pretty sure, on air plant. What that means is that these plants will hang off trees or go close to trees, but not on the soil, because they can just absorb the nutrients directly from the tree. And similarly, these parasitic plants, which are the dodder and the Indian pipe, are both non-photosynthetic parasites that either they either gain their nutrients from a host plant, or they absorb nutrients from the hyphae mycorrhizae. Either way, they're parasitic in some form. And then finally, we have carnivorous plants. Carnivorous plants, well, they most get their nutrients mostly from bugs. They live in acid bogs and other habitats where soils are poor in nitrogen, because like we said, when water goes through the soil, the nitrates are lost by leaching. And so therefore, they're going to eat organic matter in the form of insects to regain some of that nitrogen back. So you can see here, this is going to be sundew and Venus flytrap, as many of you know. And so I believe that ends our lesson for today. So we're going to have some review questions. Question 25. Which of these describes the initial interaction between rhizobium and a soybean root? So this is concepts 37.3, so we'll go back to our diagram. And I want to say that these review questions are all from Campbell Biology, 11th edition. So if we go back to the diagram here, let's see. Vesicles containing bacteria bud into cortical cells. This only happens in step 4, so step 2 rather, so that shouldn't be the initial interaction. An infection thread is initiated in the root hair of the legume. Let's look. Roots emit chemical signals that stimulate root hairs to form an infection thread. This should therefore if we look at our answer choices, it should be that the case that it should end up being the case that that is the correct answer, but I'll just make sure that that is correct. In the meantime, let's go to another question. Number 2, soil can easily become deficient in blank because these ions are negatively charged, that's our first keyword, and do not stick to negatively charged soil particles. So that means that our immediate course of action should be to identify that soil can become deficient in anions. What are anions? The negatively charged ions. So that means all we have to do is identify the negative ion from the list of nutrients we have here. Calcium, Ca2+, ammonium, NH4+, magnesium, 2Mg2+, nitrate, NO3-, yes, and potassium, K+, which is correct. Now, let's move on to the final 2 questions. And I put these together because they're both on soil pH. So let's see. How did the addition of a gene for citrate synthesis increase the tolerance of tobacco and papaya plants to high aluminum concentrations in the soil? Okay, so remember, citrate is a organic acid, so that means it's going to decrease the pH. And that means that it will increase the hydrogen ion concentration. And so it increases the tolerance of tobacco and papaya plants to high aluminum concentrations. Because of what reason? Well, remember what we said about cation exchange? Let's go back all the way up. Cation exchange is when the hydrogen ions crowd out the space necessary for attraction to occur between the plant nutrients like Ca2+ and Mg2+ to the negatively charged soil particles, right? So if citrate increases the hydrogen concentration and increases the amount of hydrogen protons within the solution, that means that the high aluminum 3 plus concentration will be mitigated because, let's see, the citric acid produced by the plant degraded the aluminum. That shouldn't be correct. The citric acid produced by the plant coated the root tips. That shouldn't be correct either. The citric acid produced by the plants bound the free aluminum in the soil, making it less available to be taken up by the plants. This seems okay. The citric acid produced by the plant dissolved the aluminum. This is wrong. And then, since all of the above are not correct, the answer must be that the citric acid produced by the plants bound the free aluminum to the soil, in the soil, and it made it less available to be taken up by the plants. And let's look at this. Why do farmers need to be concerned with the pH level of soil? Well, let's see. The pH level of soil affects water levels. This is a nonsense answer. The pH level of soil affects anion exchange. This might be correct. Plants may turn blue. This is only in a special case where we genetically modified the plants, so no. And the pH level of soil affects cation exchange. Remember, anion exchange is not possible because soil particles are negatively charged. Soil particles are negatively charged. So therefore, it must be cation exchange. So now let's move on to some optional USABO questions. These are just, for if you want to see what USABO questions look like. But, well, here they are. Question 1. Symptoms of mineral deficiency depend on the nutrient's function and mobility within the plant. In the figure shown below, a deficiency in this nutrient results in changing color of leaves to purple. What mineral is missing in the figure shown below? Well, remember our question here for the maize? Well, we can immediately identify, okay, it's going to be phosphorus deficient. And is phosphorus an answer choice? It is not. So we have to look at our reference sheet. Our reference sheet states that well, purpling of leaves will typically appear in phosphorus and in none of the above, actually. Let's see. Well, let's think about this. If we have an excess of oxygen, then that's not going to work because if there's oxygen, it will just be stunted growth. So that's not right. Let's look at nitrogen. Nitrogen is when there's a chlorosis at tips of older leaves. So that means there must be some sort of yellow. But if you look here, there is no yellowing within of the solution. However, if you look closely, you'll see that there's an increased yellowing in the middle vein. And let's look, if we look at our maze as a reference, we can clearly see that nitrogen deficient does have that sort of chlorosis in the middle, sort of in like the inner portion of the plant. So our best answer will have to be nitrogen. And if we look it up in our past USABO questions, we will find that this should indeed be correct. And we can confirm that it is, in fact, actually, it was actually potassium. So let me look at why. Sorry about that. It's potassium deficient because... I see. Okay. So since potassium has a dry tip here, it should the best answer should be potassium. So let's move on to the second question. Sorry about that. You find a wilted houseplant. In addition to the wilting, the plant displays dried out yellow streaks on the edges of its leaves. What are the most likely causes of these phenomena? Well, let's see. If it's dried out, common sense says that it cannot be overwatered, right? So let's get rid of these answers. And so let's see. The plant cell is underwater. Its cells cannot maintain sufficiently high turgor pressure. In addition, it has a blank deficiency. So let's see. It displays dried out yellow streaks on the edges of its leaves. Let's go to our reference sheet because both potassium and magnesium are both there. Modeling of older leaves with drying of leaf edges. There we go. And then this one is irrelevant now. So therefore, potassium is the deficiency that we want. And if we confirm on the line answer key, we can quickly see that this should be the correct answer. And so going back to this question, soybeans secrete chemical signals that are detected by rhizobia. No, the opposite actually. The roots. Oh, yes. Sorry, that's the right answer. Soybeans, the root will secrete chemical signals that are detected by rhizobia. That should be it for today. Thank you for watching through the entire video. I really appreciate that I took a lot of time into spending working on this video. I really appreciate any and all viewers that are watching, and so thank you for your time. And without further ado, I'll see you next time with chapter 38 of Campbell Biology.